How are specific genes expressed? Do now, what is a gene? Take a minute now and write down what a gene is. Okay. Genes are parts of DNA molecules that code for specific traits. Is that what you wrote? If not, copy down this definition as well. Notice that the picture of the chromosome is divided into strands, almost like stripes, and each of those stripes codes for different genes. You can see in this picture that there are two genes zoomed in, making a double helix of DNA. So where are the genes located? Well, genes are on chromosomes, and chromosomes are inside the nucleus of the cell. So you can see the picture starts on the right with DNA, and then wound together makes a gene. The gene winds up on the chromosome, and the chromosomes are found inside the cell in the little circle, that control center, the nucleus. Then those cells are used to make a body. What is gene expression? A human cell contains many thousands of different genes coding for many different traits. Gene expression is the activation of a gene that results in a specific protein or trait. You can see that same picture where there's a bit of a gene made up of DNA that goes onto a chromosome, which is found in the nucleus of the cell. You can see that in this cell, there are several chromosomes. A normal body cell, if you can remember how many chromosomes it has, there are 46, and in a sex cell, 23. So how are genes expressed? Well, DNA gets transcribed into RNA. Then it's translated into amino acids, which form a protein. You might remember this from the last lecture on the central dogma. The parts of DNA that code for proteins or traits are called exons, while the regions that do not code for proteins are called introns. Exons are parts of DNA that are expressed. You can remember it because the EX on the beginning of the word exons matches the EX on the word expressed. So how are genes expressed? If you look at this picture, you can see the chromosome. If we unwind the chromosome, we find all the genes. And you'll notice that the gene is made up of a double helix, the DNA ladder, and you can see that certain parts are called exons, certain parts are called introns. Those exons are the genes that are expressed. Remember when we did our last unit on fertilization and development, we talked about differentiation, where certain genes get turned on and others get turned off depending on the job of the cell. So how does environment affect heredity? Well, the characteristics of an organism actually can be described in terms of combinations of traits. So traits are inherited from mom and dad, but their expression can be modified by interactions with the environment. You can see an example of this, which is also in your packet. You can see a bunny that's all white on the right that was reared at temperatures above 30 degrees Celsius. And on the left is a picture of a bunny that was reared at 20 degrees or less. Notice that the bunny on the left has black parts on its fur. Those are because the temperature actually affects the gene that was expressed. How does environment affect heredity? An example of this includes the lack of color in completely shaded grass, even though it still possesses the genetic makeup to appear green. So you'll see those patches of kind of light-colored grass that are actually changed because of the genetic makeup in the grass. Another example is poor health habits. Poor health habits can have an adverse effect on the development and expression of many genes in human cells, resulting in sickness or even death. Examples of this include smoking, which causes lung cancer in cells. So how does each cell know what to do? Well, you might remember from the last unit, the many body cells in an individual can be very different from one another, even though they're all descended from a single cell and thus have identical genetic instructions. So you notice on the right, does anybody remember what process this is? This picture, one cell divides into two of the identical copies. Hopefully you said mitosis. Mitosis is the process where cells divide into equal parts. So these are examples of body cells, creating new identical cells. So how does each body cell know exactly what its job is if they have the same instructions? Well, it's because different parts of these instructions are used in different types of cells. This is influenced by the cell's environment and past history. Cells go through differentiation to develop a specialized function for each cell. So you can see in the picture on the left, that stem cell gets certain genes turned on, certain genes get turned off, and it creates all these different cells. So we can see red blood cells there, we can see nerve cells, white blood cells, all of the different cells that make up the body. Some more examples you can see in the picture on the right include muscle cells, liver cells, red blood cells, etc., which have a specialized job. Summary. Put the following in order from biggest to smallest. I'll give you a minute to think about this. Wow, that was a great answer. Hopefully it's right. Here's the correct order from biggest to smallest. Cell, nucleus, chromosome, gene, DNA. Did you get it right? Number 2. What's the difference between an exon and an intron of a gene? If you look at your notes, you should remember that EX should remind you of expressed. So the exon part of the gene is the part that's expressed, and the intron is the part that's not expressed. Number 3, if cells make copies of themselves, how do they know to do a different job? Hopefully you remember that cells go through a process of differentiation. This turns certain genes on to allow them to do a certain job, and the other genes that are unnecessary get turned off. Number 4, how come identical twins aren't completely identical? What else, other than their inherited genes from their parents, affects their genes? Hopefully you said the environment. Remember that the environment influences genes that are expressed,